So thank you again, Matt. And next, I would like to introduce Zeynep to present accessing CEDAR data, query, reporting, and examples. So thank you very much, Zeynep.
You see it when I switch the slides, right?
Yes, we do.
Okay, perfect. Yeah, thanks, Nina, for the nice introduction. So I will go into more detail about the CEDAR database and how to use it. So I will start with a brief introduction of how we changed IEDB to accommodate all the cancer-specific features. And then I will spend the majority on actually showing you use case examples, and then I walk you through all these examples because I think like you can learn best if you see how you can use it for actual research questions, and then we can highlight all the features that we have in CEDAR right now. I will also briefly show you how to access the data, like through a database or through downloads and programmatically, and then we'll have this very short conclusion. So by now, over the past 2 days, if you have attended, you already heard a lot about how the IEDB structures its data. We have get my pointer. So we have the reference describing an epitope, and then the epitope is being tested in the different assays. And in these assays, we have specific, we describe the specific hosts, the antigens, the effector cells, and so on. So I won't go into more detail here, but what I do want to highlight is that CEDAR uses this same underlying data structure. We kept the core structure, but then we customized it and added additional elements to be specific for cancer. When we designed CEDAR, we actually first reached out to cancer research experts to understand their needs. We wanted to know how can we make this database most useful to you, what fields do you need, what do you want to query for, and we conducted interviews and we found out we had a prioritized list, and then we worked through that. Based on that feedback, we then introduced several cancer-specific curation rules first. For example, we captured the cancer types in a more detailed way. For example, we are not just capturing lung cancer, but we capture if it's a non-small cell lung cancer, adenomous lung cancer, and so on. We also included the cancer stages when available, because this can also be very important for immunotherapy studies, for example. We also differentiate between naturally occurring animal models and vaccination contexts, and the vaccination we further categorize into prophylactic or therapeutic. So altogether, our goal was to create a search interface that enables both intuitive and granular queries, but still providing flexibility for different research needs. So this is the interface that we came up with. You'll notice it is very similar to the IEDB interface. So with several of the fields remain the same. So we have the epitope field where you can paste your epitope sequence. And then we have the assay tab, which also remains the same, where you can select between T cell, B cell, and MHC ligand assay. You can search for specific assay types. And then we have here the epitope source, which has, I guess, the most changes. Here, you can use by the gene or UniProt ID, and then you can also select these categories that we have now introduced for the cancer context. So we have the neoantigens, we have the viral antigens that are associated with cancer, and then we also have the germline and self-antigens. And then we have this little checkbox here, and if you check this, the results will also include other epitopes from the same paper, even if they're not cancer associated. So you might get data from control experiments, like for example, if they included some influenza peptides when they were testing their cancer epitopes as well. And then we have the host. This is also another field that remained the same as in IEDB. And then here in the CEDAR, we actually now also introduced the receptor tab, where you can search specifically for assays that also provide T cell receptor sequences or B cell receptor sequences. And then you can also search for specific CDR3 regions. The cancer field obviously is also very different from IEDB. We have similar, we have the disease finder here, which is you can select the different cancer types you want to include in your query. We have the stage that I mentioned before, and then the different exposure context, like if it was a naturally occurring disease, or if it was an animal model, or if there was any vaccination going on. We also have the Finder tools that you know from IEDB. They use the exact same backend. And yeah, for each of these fields, you can go into the Finder and explore them. I want to briefly show the tree for the when selecting the cancer type. So this is the disease tree, but obviously in CEDAR we will have most of the data related to the cancer node. And here we'll have all the different cancer types, and you can go into more detail. As I said, like in breast cancer as well, you can go in and then you can select like the triple negative breast cancer, the HER2 positive breast cancer, and so on. And we still will have some other diseases in here because, as I mentioned, we have those checkbox for the epitopes from the same reference that were tested. Sometimes it's a big study. They test different epitopes from different contexts, so they will also be included here because when we curate something for CEDAR, we will curate the whole paper if it has some cancer data in there. So on the right side, we have all the links to the tools that are useful for cancer research. You will hear more about these tools later from Dan as well. So now I would like to walk you through some of the use case examples I mentioned. And yeah, let's dive into it. So in this first example, let's say you're working on a literature review about a prostate cancer, and you want to find all studies that tested epitopes from a specific antigen, in this case, prostate-specific antigen or PSA. And you're also interested in identifying which regions of the PSA protein are most immunogenic. So I will show you like how to design your query. For this example specifically, because we're interested in a specific antigen, we will go into the epitope source panel, and then we will start typing PSA, and it will be the first entry that pops up here as the epitope source. So we can select this one, and under the assay panel, we check both positive and negative because we want to know the state of the data. We're interested in all the data that is available, and then everything else stays the same. So the results returns a list of epitopes tested from PSA. So we have 128 epitopes in total. And here we can see the references, the number of references for each, and the number of assays for each. So each row is a unique peptide, and I can click any of them to explore the experimental details. I will go into that in more detail later. In the antigens tab, I can confirm that all epitopes came from PSA and see how many epitopes are linked to it. Here we can also see we have a neoantigen PSA, so a mutated version of the protein, where we have one epitope reported from. So we can click on this bar chart icon here to launch the Immunome browser. And in the Immunome browser shows me where the immune responses occur along the PSA protein. The top panel shows the response frequency, and the bottom shows the number of assays at each position. Positives are shown in purple and negatives in gray. By looking at these plots, you can get a picture of hotspots of immune recognition along the PSA sequence. In this region here, for example, almost 40 positive epitope assays are reported, so this might be of interest for the researcher. Finally, I can go to the reference tab to see the full list of publications where PSA epitopes were tested. This is great, for example, for performing a literature review on this topic. All results can also be exported by clicking on this icon here, and then you can also design your custom export and select which fields you want to include in your download. So we have pre-selected like the defaults, which we recommend, but you can, if you want to make it that your table smaller or larger, you can select more or less of these fields depending on your needs. So in the second example, the researcher is studying recurrent mutations in cancer and uses prediction tools to identify potential neoepitopes. They found one peptide, for example, that is recurring in multiple patients and which was predicted to be immunogenic, whatever tools they were using. So the next question is, has this epitope already been tested? And what does the literature say about its immunogenicity? So because in this example, we're interested in a specific epitope sequence, we can just take the sequence and paste it here into the linear peptide box, and we're looking for exact matches to see what is out there. And again, we will select both positive and negative to just assess the state of the available experimental data. In the results, we see that this epitope was reported in 7 references and in 12 assays. And to look for the details of this epitope, we can also now go to the epitope details page. And you can always get to that page by clicking on the specific epitope ID here. And this opens up the epitope details page. And on the top, we have a text summary. And because this is a neoepitope, meaning it was derived from a somatic cancer mutation, we also have information about the specific variant. We see here that the protein name, where the protein change occurred, but what the DNA mutation was, and where the mutation was located in the DNA, on which chromosome and position. And we also have information about different annotations from external databases. Here, for example, we have DB-SNP, and we have the damage prediction results from external tools. And we also have the annotations from ClinVar showing the significance it was annotated with, and then also the clinical conditions. And we have also collaborated with Open Cravat. They provide many more annotations to like hundreds of external databases. So they provide this nice link here that we can go to, and they designed a specific page. So I'll just have a screenshot of the top of the page here, but this page can get very long depending on how many different annotations you have. So you will have more details about the mutation here, about the different from the different external sources. Like again, you can have information about allele frequencies, about different diseases, about the structure. So this page is always interesting to explore. So back on the epitope details page, when we scroll down, we get to this curated experimental data table. For example, we can see on the MHC ligand assays that this epitope was tested for binding to 2 different alleles, and all assays were positive. And under the T-cell assay tab, we see a total of 8 experiments, including different assay types such as cytotoxicity, interferon gamma, and then most of these assays were positive. So by examining this, the available data, this gives us a strong support that the epitope is both presented on some MHCs, and then it's also immunogenic because we have positive T-cell data. So in this last example, the user is interested in viral antigens. And the goal is to find viral epitopes that have been tested in head and neck cancer specifically, and those only with positive T-cell assay results. So to build this query, we start by, again, going into the epitope source panel, and then we select the neoantigen, and we select the germline checkboxes to only look for viral antigens. In the assay tab, again, because we're interested in only T-cell, we deselect the other ones. And in this case, we're only interested in the positive assay results. And then again, in the cancer type, we can start typing head, and to get the specific cancer type, head and neck cancer. So the results is a focus list of viral epitopes associated with head and neck cancer according to a query. And each, we have the different references and the essays again. And in this case, all of them are derived from alpha papillomavirus 9. And this is consistent with HPV-associated head and neck cancers. We can now switch also to the Assays tab to see exactly how the epitopes were tested. And this tab also includes the details like the method and the MHC restriction. We can also, again, each, like for possible for each tab, we can also export as Assays tabs now. And we get a similar pop-up. So I usually like to work with the Assays tab because it has the most detail. You can select all the different assay specificities, and then you can download it as an Excel or CSV file. So there's also multiple different ways to access the CEDAR data. What I have just demonstrated is the website where you can use just the intuitive point and click and filters, and you can explore the results, filter more, go back, change your filters. So I really like using the website for like exploring some ideas. But we also provide bulk downloads. And we also have just released our API for programmatic access. And we are also currently working on an R bioconductor package so that you can directly access and analyze the data in R. So I briefly want to show you how to access the bulk downloads. So here on the main page, we have more CEDAR. And there you can click on database export, which will bring you to this website where you have all the different subsets and different types of downloads available. For example, T-cell full includes all T-cell assay data. So this will have all the data that we have in CEDAR that were tested in T-cell assays. So we're also currently working on redesigning this downloads page to make it more intuitive. So please reach out if you have any ideas about that. So from the same menu, more CEDAR menu, you can also access the querying API that I just mentioned. And so the API, we have a really nice documentation, both in our solution center, and we also provide a intuitive Swagger annotation where you can also where you will also get help to build your specific API queries. So in just a quick summary, we have designed CEDAR specifically for cancer. We introduced new fields and query parameters to make it really useful for cancer researchers. As Matt mentioned in the talk before me, so this is all curated data. We don't have any predicted data in CEDAR as well. So our interface allows flexible queries and interactive queries. And we also provide programmatic and bulk downloads. And we are extensively linked to external resources, which is great in the cancer context because there's many very valuable resources out there. And you can get all the information you need from CEDAR, basically starting and then linking to those external resources. Okay, with that, I can finish. And please post all your questions in the Q&A, and we will have a dedicated Q&A session after the next talk.
Excellent. Thank you so much, Zeynep, for going through the query interface and providing those case studies to really try and give some concrete and realistic examples of how to use CEDAR. We are running about 10 minutes ahead of schedule, so that just means more time for the Q&A. So I'll give everyone a little bit of time to post any other questions that they're thinking of. And I will actually invite Dr. Bjoern Peters back to the virtual stage. He's very kindly both presented on day 1 and day 2, and now is joining us on day 3 for the Q&As. So thank you so much, Bjoern. And then I'll also invite Matt and Zeynep back so they can tackle some questions too. So I think maybe Bjorn, I'll let you take the first question, followed by Matt, and that should give Zeynep a little bit of time to review the questions. Thanks, Bjorn.
Sure. Thank you, Nina. Thanks everybody for the awesome presentations. And I hope I can add some useful answers. So the first answer is from an anonymous attendee. Question from an anonymous attendee who's asked for cancer-related papers. I've heard that there have been a few cases of high-profile papers being retracted. So in the case of those retracted papers, do you also remove them from the CEDAR once you're aware that they are retracted? So that is a really, really good question. It is not something we had originally thought about. And in the past, we had been occasionally notified of these kinds of papers being retracted. And then we can, in our build process, pretty much remove any paper at any time point. So we have now implemented a process where we actually, in our PubMed query, look for which papers have been retracted, and then that data is removed. The bigger issue or the parallel issue we're running into is errata. So often, very often, actually paper retractions are really rare. What happens quite more often is paper errata. Most of the time, those are authors being added to a paper. And then obviously we should be updating our author list or something like that, but it's not as important as an erratum that says the data in figure 3 was is whatever incorrect in some way or shape or form, which would require recuration. So we have to inspect that and manually and update it. And that can be actually a little bit challenging because those errata don't necessarily always make completely clear what's being still correct and what is not. So yes, our goal is to update these things as this happens. We now have an automated process implemented that does this, which I still is not live. I think is not live yet. I think we have done this, but it's still something on our to-do list, and the overall impact is relatively low. Nina, did you have something for this?
If I, yeah, just could add, that was perfect. We actually do have an article on our Discourse site that the Leidos team has kindly put together explaining the retraction process and the message a user would receive. If you think, Bjorn, I can share that in the typed responses, or I can also share my
Oh,
screen.
please do. Yeah, no. And the type responses would be great.
Perfect. Yeah, thank you.
Yeah, I will answer to related questions, I guess. So anonymous attendee asked, if the peptide was found in cancer and other pathogens or healthy, would this peptide be found in both cancer and IEDB? And the related question is, do you update the peptide classifications over time? If new studies show the peptide that was previously only found in example HIV, it is now in cancer, how do you handle this info? So just like IEDB, in CEDAR, we have all the data available. So if something was ever tested even once in cancer, it will be in CEDAR. And if there's more tests available and assays available from pathogens or in healthy donors, it will also be in IEDB. So actually, IEDB also has most of the CEDAR data as well. So IEDB is the superset of the data. And in CEDAR, we pull out the specific cancer-specific assays. So if one antigen was ever tested in a cancer context and with a T cell or B cell assay, and it has had a positive result, then we will classify it as a cancer-associated antigen, and we will also pull in assays from healthy donors, for example. Yeah. So that's why we also provide all the details about all the assays. So when you query for something and look at the results, you have the power to filter the data to your specific needs. Okay.
Awesome, Matt.
Great. So we have a question here from an anonymous attendee. What happens if you have two studies referring to the same peptide, but each uses different protein ID, e.g. UniProt reviewed and unreviewed? So this is a great question, and this comes up a lot. So whenever possible, if if the authors provide a protein ID for the peptide and it's a valid protein ID, we will use the protein ID that the authors provide. However, sometimes there's no protein ID provided by the so as Alex said, because we prefer to just present what the authors are presenting. We don't provide our own judgments. We present what the authors present. However, if a peptide isn't assigned a protein ID by the authors, or it's an invalid protein ID for whatever reason, then BLAST is our main resource for finding a protein ID. And we attempt to use high quality IDs whenever they're available, such as from a reference genome, but sometimes that's not available. And so we just do our best to find the highest quality protein ID that is available through BLAST.
Thanks, Matt.
Sometimes a peptide in the database can be assigned proteins from IDs from multiple proteins if they are assigned in multiple papers.
So let me follow up on that. I'm going to answer another question, maybe the same attendee. Which protein ID do you use? What happens if the ID is deprecated? So building on what Matt just was saying, we are putting the data in as it is reported by an author. If the author says we synthesized peptide from GenBank ID so-and-so, we would put that in even if that is deprecated in the curated source data to be able to replicate what the author actually did. Now, at the same time, as we are making our build process, we are going through all of the protein IDs that we have, say in the human proteome, and then are looking for all of the non-UniProt reference proteins and see, is there a match in the UniProt reference proteome, and there normally is. And then we group all of these records together. So if there's a deprecated record of, say, KRAS somewhere in GenBank, and that record, though, matches to the UniProt reference protein entrance for KRAS, those are then in the reporting interface that you see on the homepage. When you see the antigens listed, those are the mapped, what we call the parent proteins, where we group all of the curated proteins together. So in the curated records, when you go to the assay detail, you will find what's in the paper. But in the query and reporting interface, we are grouping things together based on this mapping to the reference proteome so that things appear in the same place. And you don't have to click through 1,000 different sequence records for KRAS. Thank you, Zeynep.
Yeah. I will answer the question from Eva. Does the database also include cancer cell lines? Yes, we do. And we're actually currently working heavily on the cancer cell lines because we want to make it possible to also search for specific cell lines. So right now you would see in the assay details if there was a cell line used or not. But we are working also on making it available that you can exclude cell lines in your query, and also for all the different fields that you can search for specific cell lines. So we're using the cell line ontology for this. For example, if you want to search for cell lines from colorectal cancer, that will be hopefully possible soon with our updated work. Okay, Bjoern.
Okay, great. We have a question from Soo-Young Kim. How big is the curation team? And Nina, correct me if I'm wrong. Currently, I believe we have 7 curators, and that's a mix of full-time and part-time curators. We also have 1 document specialist who supports us, and then also Nina, of course, as our valiant project manager.
Yeah, that's perfect, Matt. And we're really fortunate with the curation team that we do have a mix of those part-time and full-time team members, but everyone really comes together to not only curate, but peer review each other's work, make sure the curation is, a perfect representation of the paper, and gets that data out to the community as quickly as possible. So we're very grateful to our PhD level curation team. Thanks, Matt.
As they do things like present here, like Matt's just doing, but also really importantly, because that I think is the question, you're just talking about the combined curation team of IEDB and CEDAR.
Right.
The question here is we're doing the CEDAR workshop, the CEDAR dedicated curators, I think, is 2. FTEs, right, Nina? So it's not like we have 7 for CEDAR and 7 for IEDB.
Correct, Bjorn. Yep.
So the combined, and as we've been talking through, there can obviously be an overlap with certain papers. So ultimately, there's different funding pots that the money comes from, and then CEDAR pays for, I believe, 2. You should correct me. Anyway, so the size of the curation team can be somewhat hard because it's like who works on what changes over time. But that's roughly, I think, the size. Okay, so that was an extra question for me. Question from Mauricio: Is the prediction algorithms used in CEDAR the same as that used by IEDB? What do you think about the scores for selecting peptides for testing in cancer studies? So we have a combined website for all the tools in the IEDB and CEDAR, because actually many of the tools are completely identical. If you're doing a prediction of peptide MHC binding, the peptide and the MHC molecule do not know if this is a sequence relevant in cancer or infectious disease or whatever. So many of these things are completely have like a dual purpose. So we have the same tools there. Then we develop things specifically for CEDAR and cancer epitope predictions, such as identifying the typical expression level of a peptide in different tissues, because that is really important in cancer assessments. And then when we said like, okay, let's make this a CEDAR tool, people from the autoimmune community said, well, we also want to know this. We want to know which peptides are expressed in the pancreas. So it ends up that the tools can have, they're nearly all dual purpose. So we have one side and the algorithms are shared there. And the applications specifically of those can then be different. And what we are striving to do in the future is really have pipelines that then tie these tools together for specific purposes, such as selecting cancer studies, which then might use some tools that are like shared, some tools that are really just cancer specific. And that is something that we are actively working on. Zeynep.
Yeah, Dan will also go into more detail. So we have a specific talk now from Dan about the tools. So Mauricio is also asking, T-cell responses to some tumors are essentially autoimmune responses. What criteria are used to classify an epitope in IEDB versus CEDAR? For example, vitiligo and melanoma antigens can be shared. Are epitopes in these cases listed in both IEDB and CEDAR? Yes, so as I mentioned before, so whenever we have an antigen that was tested once in a cancer context and has a positive immune response, it will end up in CEDAR. And once in CEDAR, we will continue adding all the additional assays from the different, also from autoimmune assays, for example. So that's why it's important when you look at the data to also look at the assay details to see, for example, if it was a healthy host, if it was an autoimmune host where the epitope was tested. And then, yes, IEDB will have all the data as well. Okay.
Great. Got another question from an anonymous attendee. Do you have data from other resources than publications? Again, anyone else feel free to correct me if I'm wrong, but I'm pretty sure we only draw from peer-reviewed published data. Now, publications may sometime, papers might sometimes reference large data sets uploaded to other resources, other large data repositories. And in some cases, we will attempt to get that data from the repository, but we as part of our mission, I'm pretty sure, is to only use peer-reviewed published data.
I don't know, Bjorn, if you want to, the only thing I wanted to add to that is that we do have direct submissions as well. So we're funded by the National Institute of Allergy and Infectious Diseases, and as part of their programs, they have epitope identification projects, some of which I believe, Bjorn's a PI of Alex, but there's also many others, both in the B cell and T cell space. And as part of their work, they do direct submissions to the IEDB. So data that's not yet published, but has obviously been vetted, and that can be uploaded directly. And then ultimately down the line, when that data is published in a peer-reviewed publication, we do link it in the IEDB, and it goes from being a submission to a dual, where we have the submission data plus the link to the PubMed ID as well. So that's the only other, layer, I suppose.
But can I say to this again, I guess the same thing as last one. If this is the CEDAR part of the workshop, in CEDAR we don't have epitope discovery contracts that are funded that do this. So Matt's answer was for cancer publications completely correct. Now, if there ever was an NIH NCI funded effort to do epitope discovery, we would be thrilled to serve as a repository for that through direct submissions. But the normal route is, like Matt was saying, for cancer papers, pretty much solely submission. Okay. If I can answer the next question here, that's a really interesting one. Anonymous attendee asks what kind of obvious biases the data have you are fully aware of, but seems to be lost in the literature or doesn't have enough light. So that's an open question, and I would actually be curious if Zeynep also has thoughts on this, or Matt. I think the two things that come to mind to me immediately is that the mutation data is very much focused on single nucleotide variants in reference proteins. There's much less work on mutations that lead to, say, alternative splicing or fusion proteins or expression of things that are previously unexpressed completely. Sometimes that's called the dark proteome. It becomes really hard to look at those. And people just typically go for let's just go for the standard stuff, and it's probably a reasonable bias, but it clearly means that we don't have a really good assessment of the relative role of these easier to discover mutations than harder to discover mutations. The other thing that comes to my mind is the bias of studying mutations in proteins that have been known as cancer driver genes. So there is actually, when we look at raw mutation data, there isn't a big, the difference is much lower when you look at raw mutation data in terms of what happens in different proteins compared to what is then being studied in detail. It just makes more sense to study, I don't know, it is more obviously relevant to study, say, TP53 mutations than something in a protein of unknown function. So there tends to be a bias for studying things in proteins that are already known to contribute to cancer, and then that adds more papers to those studies, and then that reinforces the knowledge about these proteins, which again might not be a reflection, an accurate reflection of their overall importance or relative importance. Typically, again, just like the bias in terms of single nucleotides in regular protein coding genes, these are probably deserved biases. This is a reasonable approach to do things because you can't research everything equally, but it leads to this repetition of things that have already been studied that are being studied more. I don't know, Zeynep, if you had anything else.
Yeah, I mean, I will also go a little bit into this when I talk about the meta-analysis. But exactly what Bjoern was saying, for neoepitopes, the data is heavily dominated by KRAS and TP53, because those are the driver mutations that also not only occur in a lot of patients, but then also people, because they occur in a lot of patients, a lot of publications just go into more detail and perform follow-up assays. So it just adds to the number of assays for those specific proteins. And then also in terms of assays and what is measured, everything is very dominated by like interferon gamma, ELISpots. We have some other assays for many other cytokines, but it's only it's much lower numbers than, like, let's say, measuring interferon gamma production. Yeah.
Matt?
Yeah.
Sure. So we have another question from Mauricio. For epitope submissions, it would be helpful to provide templates with pull-down menus. Is there a plan to do this? So this is referring to, again, what Nina just talked about. We have a limited number of epitope discovery contracts with a few labs. And in those cases, those labs directly submit epitope data to the database. That's what this is referring to. And unfortunately, I do not believe there are any plans in the works to provide templates with drop-down menus. Yeah, Nina or Bjorn, you can correct me if I'm wrong, but I don't think there's currently plans in the work for that.
I can take a note though, and as Bjorn said, I had a little brain lapse, but yeah, it is for the IEDB specifically. So I will make a note, Mauricio. I know you've been working with the team, or your team's been working with our team, and I'll make a note as well, see what we can do. Thank you.
So maybe this is going to be the last question. Let's see. Anonymous attendee asks, why do you blast, not pep match? Actually, we do primarily use pep match in all of our assignment of peptides to proteins, etc. But unfortunately, I've been trying to get rid of it. The team likes to use BLAST as a shortcut for looking up peptides based on sequence identity in protein sequences. So I think, and also we use it on the website sometimes. It tends to be just easier for people to, everybody knows what BLAST is. So we should actually probably review. Sometimes our use of that term isn't 100% accurate. And I think we still do use BLAST in some cases. It's better for if you're looking, say protein homology of one protein sequence against like whatever all known bacteria or something like that. PepMatch doesn't work well for that. PepMatch works well for peptide identity in sets of protein sequences that are somewhat manageable. But again, like depending on where it's used, it might actually not be true, and we use PepMatch extensively. Is that it? I think it is.
Yeah.
Awesome. Thank you so much, Bjorn, Matt, Zeynep, for answering all of those questions. We are running just a couple of minutes ahead of schedule, but that will just give Dan a little bit of extra time. So I guess just to recap, we had Alex overview, introduce the Cancer Epitope Database and Analysis Resource. Then Matt kindly went through the curation process for CEDAR, which builds upon the IEDB process. And that was why we were able to get CEDAR up and running so quickly, because we had tried and tested curation and infrastructure processes from the IEDB of 20 plus years. So yeah, Matt went through the curation process there. And then we had Zeynep take you through the CEDAR database, the interface, all of the various query parameters. And I'm sure everyone has, recognized that the interface itself does look similar to the IEDB. So we did try and leverage that kind of brand recognition in a sense, where people who are familiar with the IEDB and were really trying to use the IEDB for cancer research, when we then pointed them to the Cancer Epitope database, the barrier to entry was a lot lower because they recognized a lot of the interface. But the key part there is that we developed the interface to make it cancer specific. So the cancer associated antigens, the disease stage, things like that, that really make it more useful to cancer researchers. And yeah, Zeynep went through some of those case study examples with us as well. And all of those are in the slides that are in the Google Drive that I shared in the chat, and I'll post that one more time. So if you do go back and test out some of those case studies, you can see for yourself as well.